Upinder Bhalla from the National Center for Biological Sciences in Bengaluru. And his research spans the structure of representations in the brain and mechanisms for their formation using both simulations and experimental approaches. And today he'll be telling us more about.
Thank you very much. It's great to be here at our parent institution. And thank you, Sandeep, and all the other speakers for making it a very exciting meeting. And especially, Munther provided a perfect intro to what I'm going to be talking about. I'm going to be talking about real neural systems, which Sridevi also discussed at some length, because they do everything that Munther was saying. They do real-time processing on extremely limited data. They make life and death decisions. They do incredibly complicated control problems, and they do it by evolution. They do it with extraordinarily high success rates. That's why we're still here. And if you ever try and cross the road, especially in Bangalore, you will, you will be testing these capabilities to the limit. Okay. So yeah, so I work here at NCBS in Bangalore. It's only about half an hour from the airport, and so do come by if you can. Okay. So what I'll talk about is a sort of a sweep, sort of breadth-first view of chemical computation in the brain. And so please don't hesitate to haul me up if I try and run away too fast. So I'll talk about chemical computation in the brain. I'll give you some examples, including memory. I'll give you a brief glimpse of how we're approaching the problem of trying to constrain this extremely complicated system. And then I'll end up with a couple of examples which I am speculating wildly about and saying, you know, these are doing things which are reminiscent of the kinds of things we see in certain kinds of modern neural networks. Okay, so this is the beast we're interested in. The brains, the 3 pounds, if you like, 1.5 kilos or so of gooey stuff between your ears, which consumes, to do all of these computations, of the order of 20 watts. And when you compare that to what our supercomputers consume, it is quite economical. Computation in the brain, contrary to what I would say a large fraction of what neuroscientists even like to believe, is not really a single domain process. It is something that spans the range from molecules all the way up to behavior. This is actually a very old concept. Terry Sijnowski and Patricia Churchland many years ago put this up, and it's been sort of there in the background, but people usually like to home in. As she was saying, there's 35,000 people come to the Society for Neuroscience meeting every year, and they all home in on one tiny facet of this very complicated system. But really, to study computation, you really need to span these levels. So I'll be looking mostly down here, but the implications really spread all the way up. Okay. Since this is a very quantitatively inclined audience, I thought I would put in the equations. I actually give these equations to the students in my basic neuro course, and they, being usually from a biology background, are usually a little bit stunned when I throw these at them. But it's good to get them started. Okay, so just quickly to work through. These equations are for chemistry. That's a chemical reaction. That's just the OD, the differential equation form of it. That is, of course, the diffusion equation. That's the Nernst equation, which relates chemical concentrations to potentials. That's the cable equation, which tells you how chemical potentials spread in a neuron and in other cables. Those two are the Hodgkin-Huxley equations. They describe the active properties of cells, what causes them to fire rather than just to propagate current passively. And that is the most commonly used approximation for synaptic transmission. That is the very crucial step of how information goes from one cell to the next. It follows this alpha function form. Okay, so this is actually just a small number of equations. And the really remarkable thing is how far you can go with this tiny set of equations in describing an incredibly wide range of what the brain does. So I'll give you some glimpses of that. But they're horrendously coupled, yes. They're all very tightly coupled. So it adds to the fun and games of modeling it and parameterizing it and so on. Yes. Okay. So just to give you some numbers, First of all, let's just give you a sense of scale, right? This is of the order of tens of centimeters. Go 100 times smaller, you get to the size of a cell. This is a Purkinje neuron. Go 100 times smaller, you're now getting to the diameter of these little branches. So it'll be tiny, it'll be about the size of this laser spot compared to that. Yeah? Go 100 times smaller, and now you're talking about what's happening within each of these connection domains, somewhat close to the molecular level. So these are the scales of length which we need to think about. Here are some numbers. There's roughly 10 to the 11 neurons in your brain. I think the closest number now is 85 billion. There's approximately that number of synapses. There's a huge number. We don't know exactly how many distinct reaction types happening in every single synapse, in every single one of those little spots, of which there's a lot of them. The electrical events are fast, of the order of a millisecond. Chemical events are slowish, of the order of a second, though note that neurotransmission is a primarily chemical process. So the signals go from one cell to the next through a chemical process, an extremely complicated chemical process. And that's of the order of a millisecond. There's structural changes. We look at those. The cells change their shape. They add and remove connections. The lifetime of a protein in all of this is only a matter of minutes to days, which is kind of scary because the lifetime of a cell is of the order of your life. And in the best case, the lifetime of a memory is also of the order of your life. And that actually poses one of the questions that I will come to. Yeah? How do you have long-term memory at all? Okay, so just some sense of the formalisms. A lot of what I'll be talking about starts with this, and this is still the domain where a lot of people work, which is simply mass action chemical kinetics. You have the binding of, say, a ligand to a receptor to form a complex. You bind something else to it to form a ternary complex. You do something else, and so on. So these are various chemical steps. This is classical G protein receptor transduction for those who really wanted to know. But of course, it happens in different regions in space. So there are compartments. You can subdivide it that way. You could do it even more correctly, incorporate space in a principled manner, say it's reaction diffusion. But you'd still be missing something important, which is that actually all of these things are happening in very tiny volumes. And so what is nice, smooth differential equations actually is taking place by individual molecules bumping into each other and sometimes reacting and sometimes not reacting. So there's a lot of chemical noise, if you like, going on under the surface. And this is profoundly important at the length scales of the synapse because there are of the order of integer, small, tens or less molecules of some of the critical chemical species in each of your synapses. So it's very noisy. Okay, so that's just a glimpse of what there is there, and these are all the things, some of the things that it does. Yeah, you transmit information, you modulate it, you adapt to overstimulation by turning down the gain and vice versa. You have all sorts of kinds of plasticity, and these are fundamental to memory. Long and short-term storage, you change excitability, you can restructure things. I don't know if you noticed that little simulation involves actual restructuring of these things called spines. You can rewire things, and based on top of this, all of this is happening at a chemical level, and they also underlie things like the pattern selectivity. How do you decide what kind of information should be retained and what should be ignored? There's spatial patterning, there's sequence discrimination, and I'll tell you a little bit about all these things. So these are all examples of brain computations which are done chemically, and I will only be able to home in on one or two of these in the talk. Okay, so this is a very, very old slide. It's something I worked on many years ago as a postdoc. Even back then, we knew a lot. This is a block diagram, and now we know enough that we can probably put some kind of a number about how much of the chemical network is represented by this block diagram. These are the important ones in the sense that they have very large effects when you mutate them. They're among the first to be discovered for this reason. But there's a lot more going on than this, and it's still a challenge to parameterize this. Just to put it in context, each one of these little blocks is a bunch of reactions, such as that. And each reaction, of course, needs a bunch of parameters to do. And this is what it used to look like when I was trying to put this together. Pre-internet days, I had to go to the library and actually pull out physical journals, Can you imagine? Younger people can't even imagine what this was like. Yeah, go to the library, take out these things, make photocopies. Ooh, how Neanderthal, whatever. Yeah. Which lines? These? Oh, the block diagram, nope. This is all purely connectivity. The hierarchy has meaning. That is, these are inputs, these are the next level of receptors, and these are the proximate computations, these are deeper down. But the lengths of the arrows don't mean anything. But the plungers mean inhibition and the arrows mean excitation, if you want to know. There's no rate information there. The rate information comes in the rate constants here. I'll come to that. It's not a silly question. It is the question. It's exactly what I'm coming to. Yeah. Okay. All right. So let's give some computational examples, and very soon we'll come to the brain. Yeah. So here's a very simple analog computation. If you have an enzyme, what's an enzyme good for? It's a catalyst, right? Yeah. And what a catalyst does is it converts many, many molecules of its substrate into its product. So an enzyme is a biological catalyst. And so having one molecule of enzyme means that you can form large numbers of a product. So this is an amplification step. And you can do all sorts of things with signs to this. You can put in inhibitory couplings and so on. So this is one example of simple computation. You have a whole range of linear, nonlinear, and other kinds of functions simply by how chemicals bind and unbind to each other. And you can have extremely sharp thresholding. You can have sublinearity. You can have logarithmic thing, whatever you like. Depending on where you're in this range, it can be different kinds of functions. So these are all straightforward things. Summation can happen in many ways. You can just have two things adding up, but there's all sorts of complicated potential computations you can do with this. Simple transforms in some sense, but you can put them together, as you shall see. Here's a slightly more complicated thing. This is a little feedback loop, one of the older, better known ones. This is something called the IP3 receptor. It's interacting with calcium, which it lets out, and then the calcium opens that. But it's got a bell-shaped curve of dependence. And so what you end up with is oscillations. So you can get huge numbers of kinds of oscillations in chemistry and in biology. And I mean, think about it, right? Your day-night cycle is just one of them. Your heartbeat is partly electrical, partly chemical. But they're longer and longer intervals. They're shorter and shorter intervals. Many of these are measured out in clocks, which are set through chemical signaling. This gentleman does not need any introduction. He pointed out that chemistry can also be used not just for temporal oscillations, but for patterns in space. And this is where anybody who is feeling sleepy will suddenly be hypnotized. But these are all Turing patterns, which are showing spatial as well as temporal oscillations. So let's get to the, I'm sorry? Yeah, so these are all simulations of chemical systems, which actually have been, some of them of Turing patterns. Yes, and some of them have actually been implemented in the, well, not test tube, but in real world situations as well. Yeah. And many of these have counterparts in biology. I mean, for example, that is vaguely reminiscent of zebra stripes, which are believed to be based in some part on Turing patterns. Zebrafish have stripes. There's even speculation that some aspects, or many aspects, for example, why do you have 5 fingers? Yeah, one of the speculations is that this is a limited length spatiotemporal wave. But I won't go there. That's not neurobiology. That's developmental biology. Okay, so let's talk about memory through chemistry. So here's a nice simple example. This is your 3-neuron organism, yeah, a 3-neuron dog. Okay, and what we're going to do is we're going to do Pavlov's experiment. Okay, so everybody knows this, right? The dog sits around. Pavlov was actually interested in digestion. He wasn't interested in memory. Yeah, so he was trying to get the salivary secretions of the dogs so that he could study their enzymatic activity. But he was a good scientist, so he noticed that the salivation went up not when the food was presented, but a bit before that, when the bell rang to tell his lab to give the food to the dogs. Yeah, and so that's a good scientist. He spotted something which was, hey, that's funny. He spotted it, and he realized that the dogs had learned that food was coming in response to a non-food signal. Yeah? They had made an association. So this is Pavlovian conditioning. And so we're going to do Pavlovian conditioning with our 3-neuron dog. Okay. And so we're going to have a bell, which is the stimulus that Pavlov used. We're going to have food. And we're going to have light as a control stimulus. Okay. And so the idea is this. Food comes in. It stimulates the salivation neuron. And so the animal salivates. All well and good? Yeah? Now, what happens is that we have a bell that comes along just before the food comes on. And so the situation then is that at the level of the synapse, which is the junction point between this cell and this cell, there's always an association. That is, this cell is active at the same time as that cell is active. Okay. And what I'm going to, the point is that this co-activation can be detected through chemical processes. Yeah. It's actually pretty well known. There's something called the NMDA receptor, which is deeply involved in this, which lets in calcium. And repeated co-activation will cause some kind of a change to take place in this synapse, in this connection. Now, so there's two things happening here. One is that there has to be a chemical process that detects that there's something salient going on, that there's an association. Because this light, for example, the lights were turned on and off, that was not associated, right? That did not get strengthened. But this association was recognized. And the other thing that has to happen is that after this trigger, after the detection of association, you have to make some kind of a change here. And that change has to be persistent. Okay, so let's spend a little time thinking about how you can do this through chemistry. Okay, how many of you know this painting? Its name, anybody? Its name is The Persistence of Memory. Okay, and the reason I love this painting is because it is exactly the opposite of what you would naively think should happen in your synapses. And there are at least 3 reasons, and I'm not really going to touch on most of them, about why synapses are a terrible place to store information. And that's where we store information. At least that's the consensus in the field. And it goes like this. Your synapses are, most of the excitatory synapses in your brain are formed on these little mushroom-shaped things called synaptic spines, dendritic spines. And they're small. This is of the order of a micron or less. And so simply putting some chemical over there and hoping it'll stay there is not going to work. It'll just diffuse out right away. So that's one reason why it's a bad place to store information. Here's another one. Yeah, I already mentioned chemical noise. If you compute the resting calcium level and count how many calcium ions there are, there's 5. 5 individual calcium ions. The number of receptors which detect signals coming in is of the order of 100. Very, very small numbers of molecules, so it's noisy as hell. This is a very, very, very stochastic environment. And this to me is the really scary one. Turnover, that is, I already mentioned, the lifetime of proteins and molecules in general in your brain is of the order of minutes to days. How do you remember anything for any length of time? So let's try and address that. Sorry, the level of what? Mm-hmm. Oh, absolutely. The cell dies. No, the cells in the brain don't replicate. Those die. I mean, there is a small amount of neurogenesis, but let's not worry about that. No. Good. So we'll come to just that. Yeah. So how can you do how can you implement such an operation through chemical means? Yep. Yeah. You have to refresh them. Perfect. Yeah. So you're getting there. We'll work our way to it very, very soon. Yeah. They don't keep alive by themselves. That's exactly it. You don't put a molecule there and say this molecule represents this memory, because it will go away. It will just be swamped by the noise. Yes. Yeah, he's incrementally. This is iterative improvement. Yeah. Okay. So let's look at it this way, right? Here's a molecule A and a molecule B, and let's just say, for the sake of example, they're in a feedback loop, and now we'll say, aha, now I know what's going on. Yeah. So they're in a feedback loop, and there's A and B in a resting form. There's it in an active form, but you have to think of them actually as a population, a pool of molecules. Yeah, and a small number of them, of the order of 10, 20. That's what I've illustrated here. So let's populate our dendritic spine, which is these ugly mushroom-shaped things I've made here. And periodically, not periodically, over time, some of them just die. Yeah, they die, they go away, and if you relied on these things to store information, yes, you'd lose it. But the cell is alive. So we have fresh synthesis of molecules, but they do not know what was there earlier. And so, but fortunately, they are, at any given instant, they're a small fraction of what there is. And so the idea is they become entrained to whatever was sitting around there. Okay, so this is the hand-waving explanation of what happens, and more technically speaking, yeah? It means that it is in a state which represented a past event. Yeah? So an active state may have represented that, oh yeah, there's this association between bell and food. Yeah? So if this synapse is active, this could be the synapse that is coupling the sound of the bell to the salivation neuron. Precisely, yeah. It's a state of activity, say, of a chemical molecule, of an enzyme. It's exactly that, yeah. Or it's the presence of a molecule versus its absence. So there is some significant evidence that this is, of course, simplistic. It's all a model, right? It's all simplistic. That I'm talking about two states. There may be various intermediate levels, yeah? But if you want an alphabet, you have an extremely rich alphabet because you have roughly 10 to the 14, 10 to the 15 of these synapses. Okay, so you can store a lot of information there. Each synapse, to first order, it's on or off. So you have either the philopodia or you have the big one. But then you have gradations in between, and we won't go there. There's a lot of stuff happening there. They're important, but I will spare you that. Okay. Yes. Yes. It is a huge question. I've spent 2 decades studying that question. Yeah. So it's actually a very, very interesting thing. And actually, Dilawar, who is sitting right here, has also looked at that problem. How do you store information for a long time using very few molecules in a very noisy environment? Yeah. So you can quiz him later. Okay. So this is basically the idea. To make it a little bit more quantitative, there's A, it activates B. There's B, it activates A. If you're a chemist, then what you do to analyze such a system is you make a dose-response curve. You say, I'm going to adjust the level of B and measure the level of A that results from that, or vice versa. I can adjust the level of A and measure the level of B. And of course, if you're a systems researcher, you know that if you now put these two steady-state curves on top of each other, which involves flipping it by 45 degrees, you can get intersection points, and those are the stable points of the system. Okay, am I going to... I usually have to spend 10 minutes on this with the biologists. I'm sorry, but this is true. Yeah, but very good. So good. So it's great to fast forward through that, but I will assume, and I'll just say that the biologists have an easy time with other parts of it. I'm actually a physicist by training, so I had a lot of trouble with the biology part earlier. Okay, so this is easy, right? So this is the stable point, and if the curves are shaped right, you can get multiple stable points. This being low activity, that being high activity, and that being a saddle node. You all know that stuff, yeah? So that's a threshold point. Call it a threshold point between these two stable states. So this is how you can implement bistability through chemistry, yeah? And through lots of analysis, which has spanned, as I said, many years of my life, we've actually found lots of systems which can do this. we've gone through lots of them. So at least at this point, there are at least 10 or 20 which have strong experimental foundations. And then in a theoretical way, you can make literally thousands and thousands of them, which we've done. Okay, so here's the one that I started out working on, a little feedback loop involving these molecules. And it has a low stable state, and you can put in perturbations, it comes back. It's got a high stable state, it will withstand perturbations. And if you're doing computation, you can always balance it like a pencil on its tip, just at that threshold point, and then a tiny, tiny perturbation will cause it to flip. So these are feedback cycles. Yeah. And they sort of emerge, they're embedded in that diagram of many, many chemical reactions. Yeah.
How can you separate when it's
Yeah, so first answer is you cannot. It is absolutely embedded. And so you have to, as in any really complicated system, you have to incrementally try and, start small, build up, figure out what are the other influences, what are the modulators that make it possible. What signals, for example, what signals cause it to turn on? I haven't even spoken about what signals cause it to turn off. Yeah, so you have to factor in all of these things. So calcium comes in through neural activity. It activates a number of pathways, including protein kinase C, which turns on something called the MAP kinase cascade, which is actually the same, at least part of this cycle is also important in cell proliferation, otherwise known as cancer. Okay, so this cycle, and then one of the many, many, many targets of MAP kinase is something called phospholipase A2, and that can in turn stimulate protein kinase C. So, and this has actually been seen experimentally in Purkinje neurons and in some other neuronal types. Something like this is happening. Another one which Delauer has studied, which was in fact proposed even earlier, there's a feedback cycle involving something called CAM kinase 2. So there's, as I said, there's a whole zoo of these potential feedback cycles storing information. So this is how, in principle, you can store information even though you lose individual molecules, right? You have a stable state, and replacement, degradation and replacement by new molecules is nothing but a small perturbation. Yeah, so you give it a strong stimulus, it will remember it for a long period. And there, as I said, many variants of it. This is one